All right. So elastin has a similar function to the elastic waistband in your underwear, in that its job is to return to shape. So an elastic waistband that stretches and then does not return to shape and snap back is not really doing its job very effectively. Your underwear is going to fall down. So that's collagen and elastin. They work together. Collagen's job is to prevent stretch and to maintain certain things. So you have collagen vascular diseases. When collagen is messed up, collagen or elastin, this is also called connective tissue disorders. There will often be a group of diseases that affect your connective tissue. For example, lupus. Lupus is an autoimmune connective tissue disease that breaks down collagen. We also have rheumatoid arthritis, which can affect the collagen in your joints. We also have scleroderma, which this lady is not angry, she just can't smile because her face is too tight. Temporal arteritis, which is a problem with the collagen of the arteries, especially the temporal artery, which is right here. If that bursts, you can bleed to death. Osteogenesis imperfecta, which is a failure in the collagen that eventually forms bone. And these children, and this is a congenital inborn defect, will be born with very brittle bones, and their bones will form badly. So you will have very strange bone defects in these children, and they will have very easily broken bones. Ehlers-Danlos is a sideshow act from the late 1800s, early 1900s, or even earlier than that, even the 1700s, as far back too. And this is, the elastin works, but the collagen does not. The collagen allows you to stretch beyond a certain point, and it will snap back into position. You will not be all loose and jowly. Now, this, by the way, these are all diseases that have like big problems, and they're not good for you. Like, they're not just cool. They're just not just neat. So Ehlers-Danlos has problems associated with it. Don't be like, oh, I want to be able to do that. No. Now, let's talk briefly about nucleic acid structure. Like I said before, you have a phosphate group, you have a pentose sugar, and you have a nitrogenous base. As you can tell, this is a lot of nitrogen right here. So that's why it's a nitrogenous base. It is a base because it has an amine group and it can accept protons, and it has a lot of nitrogen. This is what you make with it. You make DNA and you make RNA. So DNA is composed of cytosine, guanine, adenine, and thymine nucleotides. Those are the nitrogenous bases. And RNA, the nitrogenous bases, are cytosine, guanine, adenine, and uracil. Uracil replaces thymine. The only difference between thymine and uracil is one methyl group. That's it. So DNA and RNA have a lot of different structural things associated with them. We will discuss those in much more detail later. So, let us briefly describe redox reactions. So, redox reactions. This is the third type of reaction that you will need to vaguely understand for the Cambridge exam. A redox reaction. Leo-Ger. If you use oil rig, good for you. I use Leo-Ger. The loss of electrons is oxidation, and the gaining of electrons is reduction. So, when you have that, if I am losing electrons, I am undergoing oxidation. If I am losing electrons, I am more positively charged. If I am losing electrons, I am less negatively charged. If I am losing electrons, I am being oxidized. If I am losing electrons, I am a reducing agent. I am the agent of reduction. I have caused reduction. By being oxidized myself, I have reduced something else. If I am gaining electrons, I am undergoing reduction. If I am gaining electrons, I am more negatively charged. If I am gaining electrons, I am less positively charged. If I am gaining electrons, I am being reduced. If I am gaining electrons, I am an oxidizing agent. So an oxidizing agent causes oxidation in others and is itself reduced. A reducing sugar is a carbohydrate that, in an aqueous medium, generates one or more compounds containing an aldehyde group that acts as a reducing agent. Remember, a reducing agent is oxidized and causes reduction. So, remembering back from way before, which are my reducing sugars? The ones that generate an aldehyde group. That would be glucose, galactose, but not fructose. Fructose is a ketose sugar. It is a non-reducing sugar. And that brings us to, before we discuss what we use redox reactions for, the testing for carbohydrates. A solution of elemental iodine and potassium iodide, also known as Lugol's reagent, but I want you guys to call it iodine test. It changes a solution that contains starch from the color of the starch solution, which is usually clear or colorless, to dark purple black, to a dark black or a dark purple or a dark blue-black color, blue-black, in the presence of complex sugars. Remembering, of course, that complex sugars are polysaccharides, the presence of polysaccharides. The more starch you have, the darker purple you get. The color change is due to iodine interacting with the coil structure of the polysaccharide. The more coiling you have, the darker you will get. Iodine will not detect simple sugars, such as glucose or fructose. But so how will we test for glucose or fructose? We will test for glucose or fructose using, using a solution that is a weak oxidizing agent. So a weak oxidizing agent in a basic aqueous solution is capable of oxidizing aldehydes, but not strong enough to oxidize alcohols. That weak oxidizing agent is going to be Benedict's solution. Benedict's solution contains sodium citrate, sodium carbonate, and copper 2 sulfate. The copper 2 sulfate is the important part. It's what makes it blue. This is our weak oxidizing agent. When simple carbohydrates are heated in this solution, it changes from orange-red to brick-red. It changes from blue, this color blue, to an orange-red or a brick-red color. This reaction is caused by copper 2 ions being reduced to copper 1 ions by reducing sugars. Sometimes a brick-red solid copper oxide precipitates out of the solution. Complex carbohydrates such as starches do not react unless they are broken down through heating or digestion first. So, Benedict's solution will not detect non-reducing sugars. They will not detect starches. They will not detect polysaccharides. You must break down polysaccharides and non-reducing sugars first. For the purposes of the Cambridge lab, a non-reducing sugar is almost exclusively referring to sucrose, which is glucose plus fructose. You must hydrolyze sucrose. You must break it down into glucose, which is a reducing sugar, and fructose, which is not a reducing sugar, so that the reducing sugar, glucose, will react with Benedict's solution, and you will get this copper 1 ion reducing out, being reduced and precipitating out of solution. There is a color change pattern here. So from blue unchanged, you'll have no reducing sugars. In your early stages, you'll have trace amounts of reducing sugars when it turns just sort of green. Yellow to orange, yellow will be low, orange will be moderate, red or brick red will be large volume, large quantities of reducing sugars. All right? Anything greater than 4% should be a large quantity of reducing sugar. Less than that, and you will start to see different colors. Sucrose. Sucrose is a non-reducing sugar. It does not react with Benedict's reagent. It will not react with iodine because it is not a complex sugar. It is a disaccharide. When sucrose is decomposed to its components of glucose and fructose, the Benedict's test will be positive, but the starch test will still be negative. So, when you decompose its components of glucose and fructose, the Benedict's test will be positive, because it is now glucose and fructose, and Benedict's is positive with glucose, but not fructose. But there's glucose there, so it would be positive with Benedict's. Not with the starch test, because it's still not a polysaccharide. Even though you broke it down into its component parts, you keep cutting and cutting and cutting, but your hair's still not long enough. In order to hydrolyze sucrose, we have to use hot acid, but we must neutralize that acid before adding the Benedict's reagent. Why can't we just add it in? Because weak oxidizing agents, like Benedict's reagent, require basic aqueous solutions. So if we are just adding the basic aqueous solution of Benedict's reagent to hot acid, we will neutralize that, or we will have an acidic solution. It will not react appropriately. We will not have a reduction. Our redox reaction will not occur. So, testing for lipids. These tests are not typically done by Cambridge, where you basically take some oil and you test it with brown paper and see whether or not you get a spot. You have probably held some fries in a paper bag. Yeah, that's your paper bag test. Sudan red is a fat-soluble dye that stains lipids red. It is a red dye. When you add it to lipids, it diffuses through that dye. When you add it to non-polar stuff like water, it just stays as a powder and it will not disperse. It's interesting, you could actually butter one side of bread and the other side leave blank. If you sprinkle the dye on one side and you sprinkle it on the other side, you can brush it off the one side, but the red dye will actually diffuse throughout the butter on the other side. So you'll have orange butter. It tastes fine, there's nothing wrong with it. It's just a dye. Now, this is the test that Cambridge prefers. This is the ethanol emulsion test. Effectively what you do here, you take an amphipathic substance like ethanol, which is OH, carbon, carbon, there's ethanol. So we take ethanol. We take concentrated ethanol. And we use this, which has a hydroxyl group, a polar group here, and a non-polar carbon chain, hydrocarbon chain here. We use this to dissolve the hydrocarbon chains, that is the lipids. So, lipids will dissolve in alcohol, pure alcohol. But as soon as you start adding water, and you dilute the alcohol, so that it becomes more water than alcohol, the water is polar, and the lipids are non-polar. The non-polar lipids will cause the alcohol to, the non-polar water, the non-polar lipids will come out of solution with the now diluted ethanol, and you will have an emulsion of water and lipid. So you'll get this cloudy, milky, white mixture. The protein test. We take Burette's solution, which contains copper 2 sulfate again, and potassium hydroxide, KOH. Copper will react with the peptide bond in proteins that causes the color change. It will turn purple when there are long polypeptide chains present, and pink in the presence of short polypeptide chains. So, it's not going to react in the presence of amino acids because amino acids do not have peptide bonds. You require a peptide bond for the interaction between copper 2 sulfate and the peptide bond. If it's just amino acids, no bonds. No bonds, no color change. So the more bonds you have, the more deep of a color change you will get. So, this is our color synonyms chart. These are the charts that are acceptable to use in Cambridge. Burette's test will start blue or pale blue, and it will turn purple or lilac. A positive reaction that is weak will be pink. Iodine's test will be yellow, yellow-brown, or orange, and it will turn to blue-black or dark purple. It will also be blue, bruised, brown, light purple, blah. Ethanol emulsion test will start off clear and will turn milky, white, or cloudy. And the Redox test will be blue. It will change to an orange precipitate, brick red, or precipitate out copper. Other colors will be these. All right? So we are not going to interpret those colors. And that will conclude today.